Okay. Yeah, so I'll get started and we'll talk about what we're talking about. Before I do that, I'd also just like to acknowledge the traditional owners of the lands that QUT is on, which is the Turrbal and Yagara people. And so this project, I wanted to sort of start off by highlighting how it kind of fits into the center. And it is in some ways a mathsy project, but I'm trying not to make the talk mathsy, and I'm trying to link it in as much as I can. But our main goals with this was to bring this technique from machine learning known as gradient matching to the problem of population genetics. And one of the reasons also that this seemed like a worthwhile thing to do was also to sort of boost our knowledge in the area, because there's a lot of genetics work taking place in the center of different types, and we you know, we're coming at it from a very sort of mathematical background, especially starting. And so this project has been a really good way to deepen our understanding there, especially mine, at least. I can speak for myself and say that it's been, a really useful project for that purpose as well. And so, yeah, the center research themes that it fits in, because we're looking at sort of identification of selection, it comes into the genetic basis of adaptation. It relates to G2P modeling in the sense that your phenotypes then affect your fitness. And it's about looking for sort of like what, genes are being selected for in fitter organisms. And then we're also, bringing that mathematical technique or machine learning technique of gradient matching to a new context and adapting it for that context. So it sort of fits in here as well with the, new technologies. And this is the research team. I'd also just like to say that we've also had some discussions, in particular with Owen, about like some potential follow-up stuff. But these are all the people who have sort of been working closer on this work that I'll present. And before I go into what we've been doing, I'll just quickly give context to people that I have presented these ideas previously in sort of shorter time slots, and also I would say back then not having the full extent of knowledge that I have now, which is still not a full extent of knowledge, but I guess it's a bit closer. So I thought it would be good to do this seminar to sort of better represent what we've been doing and where it's come to. Yeah, so I found this image in sort of like a pretty recent paper in Plant Stuff that was just sort of summarizing different types of genetic data and sort of using that to try and understand things like selection and adaptation. And so there's 4 types of experiments here. Some of these will be familiar to some people in the center. For example, we definitely have experiments and sort of data being collected for fitness, or at least, sort of like the phenotype that emerges for plants that have been grown in a different environment. There's also sort of like the genetic differences that you see across an environment. You could kind of look at the Senecio stuff with June and Headland as maybe like an example of this, where you just have 2 of these points and you're comparing those, obviously doing it in a more thorough way than just looking at allele frequency, but that kind of fits into this. There's also these ones at the top where you're looking over generations. The first one is where you kind of pick, I believe what they are suggesting here is you're sort of enforcing a type of selection yourself and then seeing what genetic difference emerges when you enforce that selection over generations. Or there's the one where you actually measure the frequency in generations that may be experimental or might even be in the wild. And the point is you're just looking for how an allele frequency changes over time. And that's the type that we're talking about today, this type of data. And so those types of experiments, what you're really looking at is, well, if I have this example here, that allele frequency looks like it's going up. So maybe that means that that allele is being selected for because it's, having some benefit to fitness and it's advantaged in an evolutionary sense. So that's the sort of data that we're talking about. And we're looking at a way to try and identify what's going on in this type of data is what the project is essentially about. And so this fitness, actually, I'll talk about that later on this slide. Okay, so there's sort of two ways that I could see this being used, and this is kind of more of an if, because as I said, we're kind of bringing the method for how we suggest doing this. So we've adapted the method to this context. We're seeing if it looks like it's a useful method in this context. Then the question is, well, how do I go and use that? The two ways that seem most obvious when you have a gene that you already are interested in and you want to see if selection is acting on it. And sometimes that can just be because you can see the phenotype. So the example of the moths that we'll talk about, if they have a certain gene, it changes their coloration on their wings. So you can have examples where, like a sort of a single genetic site is controlling one small trait. And if that's observable, then you don't even need to do your sequencing to collect allele frequency data. You just collect, the plants or the animals and track their phenotype as a way of seeing what alleles they have. And so when you're doing it in this sense, you're really looking at, well, I have good data for this, or hopefully good data, and I want to see, is selection happening here? What kind of selection? Or maybe it's drift, and we'll come back to that point. But the second way that I've seen people using these types of methods in the literature is when you're detecting signals of selection. So what I mean by that is you're looking at like a whole lot of loci, and you want to say, well, which ones of these do I think are being selected on? So I couldn't tell you much more about how pool-seq in particular works, but that's the sort of data that people have been talking about when they're doing it in this context here. And I suppose you can kind of think of this like a GWAS type thing, except instead of trying to do your G to P, so your genetics that determine the phenotype, now you're kind of looking at, well, does this one look like it's being selected on? Does this look like it contributes to fitness? So it's kind of a G to fitness type version of GWAS is maybe how you could look at this. And just like GWAS, a lot of the methods will just do like an individual check on each site. And then when you see ones that are close together, you'll see that they all kind of have some signal because of genetic linkage. Sites that are next to each other tend to be inherited together. But you can still, throw your sort of statistical methods at that data and say, yes, this one looks like it contributes to the trait in GWAS. Or in this case, yes, this looks like it's got a contribution to fitness because it's being selected for or against. And so those methods are just sort of looking at lots and lots of loci, lots of data, and trying to pick out the ones that they think are, potentially being selected on. That's probably all we need to say about that. Okay, so I don't really want to make it like an interactive session for like a recorded webinar, but what I want people to do is to just look at this and think to themselves, okay, I have an allele frequency for this allele in these moths that controls, like a certain patterning of the cream dots on their wings. So they were observed over time, generation to generation, a lot of years of data, and they saw it decrease quite sharply in frequency of the mutant allele that changes the cream dots. Maybe it kind of recovered a bit there. I'll also just quickly note that there are zeros here, but zero doesn't necessarily mean zero because you don't know that you've caught all the moths, obviously, at the site. You're kind of just getting a sample and using that as your, suggestion of what the allele frequency is. So these zeros aren't true zero, and it doesn't mean it's necessarily fixed or anything like that. But yeah, we started with a high frequency. It came way down, hit what looks to be close to zero, maybe bounced back. So the question I want people to think about to themselves for, like 20 seconds before we move on is just, would you say that was selection? Has there been, like, does changing those cream dots really make a difference to the fitness of the moth? the only things I would add while you're thinking about this is like, well, it's decreased quite sharply. That looks like something's going on there. But we also have to be aware that a lot of alleles may appear selected because of random fluctuations and drift that just happen to go one way. Because especially when you're looking at lots of genes, some of them will look like they're doing a consistent thing, even just randomly because of pure luck. So the answer is no one knows. It was kind of a trick question. I wanted to make the point that it's actually very difficult to understand whether this is definitely being selected on or not. And there was actually arguments in the literature way back here, and it's sort of continued on in spirit, these arguments between, well, mostly we're just seeing drift, most alleles are neutral for fitness, and then the opposite camp, which is there's definitely selection here, and there's definitely a lot of selection going on. In terms of the sort of mathematicians who were sort of basically having this fight in the literature, you're looking at Fisher, who was on the side of selection, and Wright, who was on the side of most alleles being neutral and it being mostly drift that we see. So yeah, it's actually very hard to tell them apart. There's no sort of easy one answer. And especially if you're doing multiple tests, you may see something that is just drift, but it looks like selection. It's because you've checked lots of sites. So, you picked the one site where you had, luckily or unluckily, it's just been going down, down quite consistently, even though that was just the effects of random mating and just random genetic drift. So yeah, it's very difficult to sort of tell these two apart. And that will be kind of one of the main themes of what we're going to be talking about. And the other things that are challenges in this space is, firstly, everything that we'll be talking about here is at a single site. And so you can sort of repair that by saying, well, I'm just checking for a signal, the same way that, GWAS can look for signals despite not taking account of everything that's going on with linkage and recombination. And the second issue is, if I want to do those big, searching for signals of selection, I need a very fast method. And the mathematics that tries to pick out what it thinks is going on here by default is not fast. So those are the two that we'll be dealing with today, is the speed, and then also, as I said, telling apart selection and drift. I'm just going to quickly check time. Yep, it's okay. Okay, so I haven't put a ton of the mathematics in here, but I wanted to talk a little bit just about what our model is for what's going on. And so the basic model that's kind of fundamental and treated as, the model in a lot of these studies, when you have non-overlapping generations in particular, which is often the case for plants, so that's kind of beneficial for us in that sense. You have the model, which is the Wright-Fisher model. And so those are those two authors that I said were continuously arguing. They're both kind of credited for use of this model. And what it says, I've included the version with selection in here, and we'll talk about that, is just that the allele frequencies, which we've called X at generation N, are a multinomial sampling of the allele frequencies at the previous generation with selection having acted on them. But if there was no selection, then this would just be X in here. And so what it's really saying is the allele frequencies you expect at the next generation are going to be whatever the effects of selection are, but put a bunch of randomness in the process. And that randomness comes from, like which sets of genes pair up with which sets of genes in reproduction. So it's a random sampling process because you're essentially picking like who are the parents and then what genes get produced from the parents into the next generation. And so that's where you get a sort of randomness effect. And that randomness allows the possibility of drift where, oh, well, it just turned out that the genes that appeared in the next generation were more of those ones and less of those ones. Not because of any selection effect, just because those are the parents that decided to mate randomly, or, those were the genes that were passed down through reproduction. That's kind of the model here. The only other thing I'll say is you'll often see this with the 2n. The 2n here and here is because if n is the number of organisms, then 2n will be the number of alleles if they're diploids. So here, when I say n, it's the number of alleles if you're doing this as a model for alleles. And so, yeah, this function that I've sort of hinted at here is just saying, well, what frequencies of alleles do I expect if these are my previous ones, and I do have some sort of fitness acting on there. So we'll talk more about that later. The other thing I'll just quickly say, this expected proportion that I've been referring it to, you'll only actually see that in an infinite population. Because if you have an infinite population, now your randomness of mating kind of averages out across infinity, numbers of individuals that are all doing the same process. In statistics, it's kind of like a law of large numbers effect. And in a previous talk, I did sort of show some sampling examples that just showed, if you have 12 individuals in this, you can get all kinds of results. But if you have 12 million, you're going to see frequencies that come out pretty similar to what you expect. And as you go to infinity, that, just gets even closer and closer. Okay, so we don't need to talk too much in detail about the selection function and this normalization function, but I just want to say the main point, which is that selection acts by saying, well, if I have higher fitness, I'm more likely to appear in the next generation. But also, if I'm currently an allele that's frequently in the population, I'm more likely to show up. And so the way that we get a selection function is we say the alleles that are there are more likely, and we weight also those current frequencies by how fit they are. This circle thing is just the element-wise product. So it's like the frequency of the first allele with how fit the first allele is. That's my weighted measure of how likely I think the first allele is to appear, and then so on for all of the second and third, or however many alleles there are at the locus. Often it will just be 2. Yeah, so that's how we get selection acting to move us to the next generation. We don't really need to talk about the normalization function thing here. But you put it in there just so that you get probabilities, because remember, these are our sort of like our expected fractions in the next population. In an infinite population, this is what we would see. So we need a function there to make sure they're actually adding up to 1 and they represent proper probabilities to go in that multinomial. Okay, so when we're trying to learn selection, what we're really trying to learn is fitness. And the methods that we use here are learning frequency-dependent fitness. So you have this fitness, which is like your phi symbol here. But we're saying that actually depends on x as well. And so when I say fitness depends on frequency, that might seem a bit esoteric, because a lot of times when people think about fitness, they're thinking about, oh, well, this allele, creates some sort of phenotypic difference that is beneficial for the plant or for the animal, or for the bacteria, whatever it is that we're looking at. So in that sense, you might not be thinking, well, why is there a frequency dependence? So the maths looks at it as a frequency dependence, but that could mean all kinds of different things. For example, even if you just have a diploid population and your individual allele is just doing like some phenotypic effect that has a constant effect, it doesn't depend on anything what else is going on in the population, you will still actually get a frequency dependence in how you describe that mathematically. And the reason for that is, if I have, for example, a population that's 99% wild type and 1% mutant for that allele, well, then that means that I'm going to have very, very few homozygotes of the mutant, because if only 1% of the alleles in the whole population are mutant, then what's the chance that I get one individual plant that has, 2 mutants at the site in a diploid plant? So as soon as you go to diploid organisms, when you look at the fitness of the individual alleles, it will actually depend on the frequency, how many of those alleles versus the other alleles there are in the population, because it will change how many heterozygotes versus homozygotes you have. and the heterozygotes and the homozygotes may be differentially fit. So now, yeah, your fitness actually matters depending on the frequency. But there's also other examples where this applies. You can look at oddity or dilution effects, and this is where you can have a fish in this example, but it happens in a lot of different cases. And by looking a different color, it may stand out to the predator. And so the predators will sort of prefer to target the ones that they can track better. It's very common in swarming populations. The thing, that stands out will be the thing that gets predated more likely. But you also have the opposite. You can have an effect where the predator has sort of trained itself to hunt its prey. And so the prey that look or act differently are then actually favored. The predator won't prefer to go for them over others because they don't look like what it's used to predating on. So you can have both types of effect. It's just sort of like what population you're in. But both of those will create a frequency dependence on the fitness. Because if my mutant turns me white, and I've got lots of white fish because I've got lots of mutants, in which case maybe it's not a mutant, it's actually the wild type. But the point is, if allele 2 is white and allele 1 is red, allele 2 in the fish example is really hurting me because I'm standing out, and these fish are one where the oddities get predated on more likely. But in a population that's mostly white, now the red fish are the ones that are going to be differentially targeted by the predator. So you can see that the current makeup of the population, how much white versus how much red, will affect how fit the alleles for white and red actually are. Another example that was pointed out to me in a previous time when I presented this work was self-incompatibility, which I've seen some papers about that look into the genetic basis of it, and I get the feeling it's a lot more complicated than I'm going to be able to explain, let's say. But the basic idea is because you have a lot of plants, especially flowering plants, that sort of represent both sex organs, they have mechanisms built in to sort of avoid fertilizing themselves to sort of keep up genetic diversity, essentially. And so if you have that type of behavior going on in your system, then that is going to create a frequency dependence for fitness as well. Because if you have alike alleles for an allele that is one of the ones that's sort of doing the self-incompatibility, because there's like certain genetic bases for it and like sites that kind of control it, then that will create a frequency dependent fitness effect as well. And the last example is that because we're using the Wright-Fisher model and then we're converting that to the replicator, which we'll talk about a little bit, but not in detail as we go on, the replicator is actually used a lot in evolutionary games. And so it turns out that a lot of our theory and methodology that we use here will also actually apply in evolutionary games. What are we talking about with evolutionary games? Well, there's a whole lot. You can think of like the prisoner's dilemma and stuff. But there's also things like, for example, hawk-dove game. And that will be where if you choose to be aggressive and you fight, you encounter lots of other members of your population that choose to be aggressive, you'll just end up fighting the whole time, and it will be sort of detrimental. If you have a mixture of aggressive and peaceful individuals in the population, well, then the aggressive ones will be winning out because they can kind of dominate the peaceful ones and take the food or whatever it is. Or you can have a completely peaceful one, in which case, the peaceful one is sort of winning out without the competition. So depending on how much aggression there is in the population will affect how good it is to choose aggression as your strategy. Anyway, we talked quite a lot on that slide, but I think it's just because it's interesting to see the different ways that frequency-dependent fitness can emerge. And one of the key things is a lot of the methods that we're comparing to in the paper we've been writing for this are focused on this case, because they are often assuming a basic model of constant fitness, but then we get some frequency dependence from the fact that we're dealing with diploids, and we're trying to estimate the parameters of that known model. But our approach, as we will see, is kind of generally applicable for any frequency dependence. So we can try to infer what's going on, at the sort of allele fitness level with these other types of dynamics as well. Okay. So the way that people do this, the way that people look at that frequency data that we showed on the first slide and said, okay, I've got data over generations. What does that tell me about whether I think there's selection going on or not? The way that people use that data, if you want to use the most basic method, not basic as in easy, but basic as in it's fundamental, you would go to that Wright-Fisher model, and the mathematical details don't matter, so I sort of, blanked it over here. But so you go to that model that I talked about before and you say, okay, what are the chances that I saw that data, assuming some kind of pattern of selection in here and, the randomness of mating for some population size. And I may be trying to infer the population size as well, or maybe I know it, or maybe I've used a different method to estimate it. But yeah, so I could try different patterns of selection, see how likely it was that I saw that data under this model, and then pick out the one that was most likely to have generated the data. And that fundamentally, statistically, all makes perfect sense. The problem is it's very expensive to do. And so there's been a bunch of other methods that have sort of emerged that try to avoid that, basically. One way you can do it is you can look at, under certain assumptions on this model, you get other versions of the model, or you can look at the moments of the model, which is like the overall mean behavior, but also the sort of covariance, and you can try to match those, or you can approximate with diffusion and then maximize the likelihood under your diffusion model. So you can kind of avoid the expensive model, but still get the same effect of saying, well, if those assumptions I made are all okay, what were the most likely selection effects I could put in here that would give me the data? Another way you can do it, which is the approximate Bayesian computation way, you may have heard, like statistical papers talking about ABC methods. That's what we're talking about. What you do is you just pick lots and lots of trajectories that came from what you believe the model is for some selection here. You try lots of different selections here, and you're going to generate lots and lots and lots of trajectories. And then you're going to compare which ones look similar to the data. And then obviously the ones that look close to the data, you're going to say, oh, those are the ones that are plausible. Those are the ones that I believe may have matched selection dynamics. And the nice thing about this is, yes, there's random luck in what, random mating takes place, what random numbers you generate when you sample the multinomial in here. But if you do this lots and lots and lots and lots and lots, you can pick out the ones that only do it very rarely, come close to the data, versus the ones that come close to the data more frequently, and say, oh, I think those are the more likely ones, but the other models, there was a chance of those. And so you get this kind of uncertainty about what your answer is, but you still get beliefs about what you think might be going on. So that's sort of the most natural Bayesian way to do this. But as I said, that's lots and lots and lots of sampling of this to do it sort of thoroughly and properly. What we do instead is we say we want to do gradient matching. And to do gradient matching, we need gradients. So we look for a differential equation, and that's where we come to the replicator, which can be sort of derived from evolutionary processes under certain limits. The main one is that you're going from discrete generations to continuous generations, and that you're kind of removing the randomness part that comes from drift. So you're assuming an infinite population, essentially. And so the bad thing about this approach is there's no n in here anymore. As I said, it kind of assumes n is infinity and just disappears from the maths. So we can't use this approach to try and figure out what we think the population size is, or the effective population size. But it turns out to be useful for other reasons. And it's the wrong model in the sense that there's no drift in here. This is just a completely deterministic, which means like you know what you're going to get every time you run the model. If you pick some fitness to put in here, you're always going to get the same answer. So we have the wrong model. There's no drift. We don't even think about random chances, but that turns out to sometimes be a benefit. And so if we're using the replicator, we can get sort of two approaches for it. We can do it with nonlinear least squares, where we just sort of match the trajectory to the data and say, did it match? Or we can do this gradient matching idea. Okay, so to show the gradient matching idea, what we're talking about is looking at the slopes versus looking at the trajectory. So in traditional sense, I'd say, oh, if I picked this fitness, I would get this curve. If I pick this fitness, I get this curve. If I pick this one, I get this curve. Okay, what's kind of the best fit to the data? In this case, out of those three options, we would pick the green. So that's the trajectory sense of matching. which is just nonlinear least squares if you want to use the mathematical terminology. But the other way we can do it is we can look at the slopes through the data. So we don't have slopes here, but we can estimate them. We'll talk briefly about that. But if we do estimate the slopes, now we're trying to match what do these different models predict about the rate of change of allele frequency at the different points in the data. And so now we're looking at the arrows and we're comparing the arrows that match. And if we estimate the data well and the slopes tell us the same story, you're going to still be picking the same model. Like the green arrows will still be coming out closer to the data than those that represent the blue or the light blue. I did just draw this in PowerPoint as a sort of illustrative example though, so don't take, the slopes of these as faithful recreations of this. It's more just sort of a demonstrated image. Okay, so what are the benefits of this? Well, in machine learning, a lot of people use it because it's faster. Because if I don't have to solve this equation back here, I can just say, well, the slope, which is dx dt, is whatever the slope on the right-hand side is. So if you tell me what the fitness you think it is, I can just tell you what slopes that predicts and then compare them to the slopes in the data. So in that sense, I never actually have to solve this equation. And, solving a differential equation can be expensive to do it accurately. And the other thing is it actually comes out with simpler problems, but we'll see that visually later. So in the interest of time, that's all I'll say for that now. But the other benefits that seem more specific to the genetic problem is because we need to take discrete data from generations and estimate it with a slope through it to get what we think these arrows are, it means that we have to smooth the data inherently. So we actually are smoothing the data in a way that kind of comes with the method, as opposed to just saying, well, that was the data, but I'm going to smooth it as a correction step to make things better. Instead, it's kind of, well, we need to estimate the slope to use this method. So it comes kind of baked in with the method. And reducing the noise by smoothing the data can help with identifying selection. And the second thing is trajectory free. So here we have to follow the trajectory, right? So if we had the right type of dynamics predicted, but we ended off at the start because we were misled by a little bit of data at the start, we may get a trajectory that looks really bad, even though it's doing a very close to the correct thing in terms of selection. Our notion of error is accumulating because every time we step through that equation and integrate it in order to plot out what we think this, this population will do under this type of selection, we're kind of building up error. If we do it this way, each individual slope at each point, just contributes to the matching equally. And there's no build up of anything because we're not integrating the system. We're not adding it up over time to plot the trajectory. Okay, so to recap the method, so far, you've got your SNP data here. You work out what you think the slope through it will be, and then we're going to use that to work out what we think the selective pressure is. So S and H are like parameters in a genetics model, or maybe we're just trying to learn what we think the frequency dependent, function is overall. And so because we're using this gradient matching approach and it makes things simpler in a lot of ways, we do get an advantage in that we can go a bit further and try and learn a general model. And that's why this approach is better applicable to all those different cases as opposed to just a known model. And the way we do that is we say, well, let's just make our rate of change equal to the sum of some pieces of function. And we're going to just pick all the different pieces of function we might think go in there. And then we're going to learn the parameters in front of those. And that will give us like a very flexible type of dynamics we can learn. And you may notice, well, we'll talk about that later. But yeah, we may notice the issue here, which is, oh, if this is allele frequencies and I just add on all these random functions here, how am I going to be guaranteed that I still have proportions that add up to 1 so that they actually represent frequencies in the population? I can't just add up some function pieces in general and say, oh, well, now I've got 13% of allele 1 and 112% of allele 2, because obviously that makes no sense. I need to keep everything as proportions. And so what we did instead was we said, we're going to take the idea of equation learning, but we're going to make it a fitness learning approach. So in that replicator equation, where we have fitness, we now replace fitness with a bunch of pieces of fitness functions that, are from some library of options. And we're just going to pick what options we think are actually appearing in this problem. So like what selection do we think is going on out of all the options of selection that we could have? And we're going to do that by matching the slopes estimated in the data. And one nice thing here, I won't get into the technicalities of it, but this is linear, and in mathematics, linear is always good. It's linear in terms of the thetas, at least. The dynamics in terms of the fitness functions may not be linear, and the dynamics in terms of what the allele frequency does over time is not linear in any sense, but it's linear in the thetas, so we can actually estimate for the thetas with linear system methods when we do gradient matching, which is nice because linear methods are really fast and you can do sparse regression easier and things like that. Okay, so I don't want to talk too much about this in the interest of time, but this is basically just showing what an example fitness library will look like. So these are all our pieces. You can see, the fitness of the first allele, which will be the top row, and that can be a constant term for the allele, fitness of the second allele, and then you can have the dependence of, well, how many X1s are in my population? How many X2s are in my population? I'm going to let that linearly affect fitness as well. And if you do that, it actually simplifies down to learning a payoff matrix, which is the evolutionary gains way of saying, I have a matrix that describes my frequency dependence I'm trying to learn. And that's perfect for our purposes because a lot of the known models, for example, when you just do the diploids known model I was talking about before, you just have selection strength and dominance in the heterozygote, and those parameters naturally fit into a matrix form like this to represent your frequency dependence of your allele fitness. Okay, so it fits into this form is really what I'm saying. Okay, so how do we estimate the gradients? How do we get this dashed line through our data that gives us an estimate of like what is the rate of change of allele frequency over time? And so we've used Gaussian processes for that. The important things to say about it without getting technical are the things on the slide. Most notably is like, it doesn't matter what the form of this is. I don't need to specify what I believe. It's not like I'm trying to learn the best fit straight line through the data or the quadratic curve. I'm not specifying any fit. This is just some random bumpy function that follows the data in a smooth way. So that's why it's a nice method for our purposes. There is a technical thing we have to do though, because we're going to fit one of these slope generating processes to all of our different allele frequencies, especially when we go to multiple alleles, then it's not just going to be X1 and 1 minus X1 anymore. We do need to do this like projection step so that we can make sure that this slope function that we predict keeps the alleles as genuine frequencies throughout. Okay, so recapping, we just have an extra sort of step from what I did in my previous recap. Now we take our, our sequencing data, we pick one allele at a time, we figure out what the slope is through it. That slope gives us estimates of what we think the rate of change is, and then we match that to our fitness library function, and we work out what are the best values of these coefficients theta that will give you a match between left and right-hand side. And those thetas, they may equate to the payoff matrix. If it's symmetric, we can read out S and H, or we may just be learning an arbitrary fitness for an unknown model, which is those other examples like, do I have an oddity effect going on here? I don't know. Let's have a look and see what we think kind of thing. Okay, so now we'll just jump into the results. The main things that I want to highlight in the results is this first example. So as I said before, the Wright-Fisher model is the kind of fundamental model in the space. But if we give it a very simple problem where we just generate data in a finite population, it's noisy, we have no selection going on, so it's a neutral allele, the Wright-Fisher model, maximum likelihood, will actually say, no, I think it's a very strongly selected in one direction allele. It's negative here, but that just means, the other allele is favored. And I think the heterozygote is even more favored because HS is now switched around to be a positive value, even though S is negative. So that means that H is like very negative. You have very strong underdominance, which is the same as saying the favored one is very, it's overdominance for the favored one. But what that's saying, let's look at it graphically, is, well, you're giving me this model that just generates random trajectories. There's randomness of mating in there. I want to keep it close to neutral data. The allele frequency isn't really changing over time beyond the random noise. What's the best model to do that? And you might say, well, no selection should be a good model, right? Because that's what's going on physically. But if you generate Wright-Fisher trajectories under no selection, you get this. And what the mathematics, cleverly or dumbly, you decide, does is say, well, if I had really strong selection that pushed me towards this value right here, even though I haven't seen data from anywhere else, so I have no idea if that's going on, that really strong selection will keep my trajectories closer to the data. And so this model where I've introduced really strong balancing selection to this frequency right here is, 10,000 times more likely than this one, or whatever the value is that I figured out recently. It was like, yes, 4 or 5 orders of magnitude, I think it turned out to be. So the point is, we learn this very overconfident model that suggests we have very strong selection acting here because it gives us better likelihood when we take into account drift. And so when we use our method that ignores drift, we don't fall into that pitfall. And especially the gradient matching version, we actually get, much better performance even when we have the difficult case where we have no selection and randomness is liable to, mislead us. The interquartile range, so like the middle set of results, is very, very good for the challenging case, showing that we're getting a lot less variance in our predictions and we're centering them on the right place, which is great. Wright-Fisher gets it wrong. And if you do the replicator version, but you use least squares instead of the gradient matching approach, it still does a lot better than Wright-Fisher because it doesn't get misled by this effect, but it does struggle a bit more. So gradient matching with the smoothing and all those other benefits is helping us as well. This figure just shows that you get more complicated surfaces. So when we try and find the maximum likelihood, we have to search the whole space of what we think selection might be doing and find the one that's most likely. And so when you have this weird looking surface with like a ridge here, and you can see these little lines that show the arrows of direction of descent, they're pointing in random directions in some places, and you've got these weird places where they're pointing away from where you want to go. Here, you get much more consistency. You only get a little bit around here, and it's just because it's a very steep ridge, and the way I've plotted it kind of doesn't show that. And there's a better example of that after. So yeah, identifying that the replicator model looks promising, we sort of then did more thorough testing across different population sizes. We tried the case where you give it the true model in this case. So this was just diploid selection. We know the true model is that symmetric one with the S and H, but we tried giving it just general polynomial libraries like I showed, not constrained to be symmetric. And in terms of predicting the correct trajectory of selection, it still did, basically equally well to knowing the true model. So even when we don't know the true model, we can at least still identify the right trajectory of selection that's going on underneath the noisy data, or at least equally well, because it's always going to be challenging to tell the two apart. And if we were looking at parameter estimation, you see the expected thing, which is as you make your population bigger and you see less drift, you get much better estimation of the parameters and much more confidently. But we still do see, compare blue and red, it's slightly improved in the tougher ones when you look at gradient matching, which is our kind of novel contribution. Okay, so there's just a few more results to sort of finish us off. I'll just talk about them very quickly so we have at least 10 minutes for questions. This is where we now have 3 alleles at the site. So a lot of the time you just talk about a mutant versus the standard wild type allele at a site, but you do have other cases where you can have multiple alleles that feature at one site. And so here we're plotting it on a triangle, which is just kind of representing that everything has to add up to 1. So if I have lots of x3, well, then I'm very far away from x1 and x2. So this is like all x3. This dot in the middle would be the population of every allele appears equally frequently. And so, yeah, we just came up with a few selective trajectories that showed different things and tried to match them. In this case, you can see both gradient matching and the standard replicator fit are still misled by drift. The true selective trajectory is doing this, but the drift kind of just went down here. And when you have data that's sort of that clear, we're not going to just magically ignore the drift and pick out this trajectory. The benefits that we get from gradient matching more come from not getting misled by trajectory. And you can see an example of that here, where the drift kind of came away and then came back. The trajectory matching one wants to make sure it goes through the points here. It's a bit hard to see, but the red line is going through the points. The blue one is looking at the slopes that are going on, and it more faithfully follows the gray for a longer amount of time. But again, when you have noisy data, it's not going to give you a perfect fit, especially when you go to more alleles. As you increase the population, you get a lot less noise, then you do get much better agreement, of course. This is just showing box plots for those different cases. The main things that I'd want to point out is just that gradient matching, so blue versus red, is going to be essentially equivalent or better in almost all cases. And in the cases where you really need it, the transient one, which is the hard one, is the one where we see the biggest benefits for red versus blue. So that's where gradient matching is really benefiting us. And actually, transient one was this one that I said is, tough. And here in the example that's randomly picked, we failed. But overall, especially when you increase the population, you are getting much better performance with the gradient matching version. Okay, and that's parameter estimation. We won't talk about the details of that. But again, compare the red and blue, you do see like much lower variance in your estimates, which is very good because it means you're more consistently giving answers close to the true one, which is the black line. And when you have the type 2 one where it works a bit better, you still see that blue is pushing it towards the line better. And where it fails in these cases, it's like more of an identifiability issue. It's the data doesn't really tell you enough about the full scope of what's out there. Because, like if an allele never really shows up in high numbers in the data, you're never going to really know much about what that allele is doing. So you have some flexibility in what the true model can be, and it picks one that's slightly wrong. The last example, actually these results are potentially needing to change based on something I discovered sort of like yesterday-ish, which means I don't want to talk too much about them, and also in the interest of time. But the main thing I wanted to show here, which is a fundamental benefit of our gradient matching approach, is that we're more able to pick out the data in the complicated case. So here we're looking at a rock, paper, scissors game. If you've got lots of people playing rock, then scissors will be bad and paper will be good. But then people will start picking paper, which will mean people will be now looking at scissors. So it creates like cyclic dynamics where what's favored in the population keeps changing based on what people are choosing to play. And you can see this in some bacterial and lizard populations as well. It's not like a hypothetical exercise. But the point is, if we use gradient matching with the blue, we're able to pick up that cyclic nature a lot better than the red. And the reason is the red is trying to fit this trajectory, and it's a very difficult way to find that trajectory because the way that you measure your trajectory error comes out just in a very complicated way that's hard to match with cyclic data. And the best way to show that, this is just showing, that was one example, but if I showed 10 examples, we're still doing much better than the red, which is all over the place at picking out the cyclic nature. So you look at this example, you can see why. If we use our gradient matching problem and I show like a slice through the full parameter space, we see that it's very nicely pushing us towards the true likelihood. Well, the true one is the red and the optimized one from the data is blue. But yeah, like they're both close and we're pushed towards that point. If you look at this case though, which is if we just do the trajectory-based nonlinear least squares one, we're all over the shop. If I start close, I probably will end up in this spot here, following this kind of ridge down. But if I end up over here, for example, I'm actually being pushed away from the right spot. So you can see that if I try and follow the slope down to find my optimum, which is how these methods tend to work, I'm going to be misled if I don't have a good initial guess. So it's a very much harder problem to optimize, and that causes bad fitting. So this last result, I'll just be very quick about. If we fit our method to this data, we actually found that the gradient matching one, it picks a model where the cream dot is slightly less favored. And that's a far cry from what other methods in the literature and also our nonlinear least squares one picks, which is if you get double mutant for the cream dot, you're just dead. Like there's no appearance in the next generation. And if you're the heterozygote, well, you're roughly as favored. So you can see that the models are actually quite similar because double mutants are very rare, but the model that ours picked is a lot more believable because there's no real reason that having double mutant kills you or makes you, completely sexually useless and you can't appear in the next generation. Because they have observed, the double mutants rarely, and it's not like they can't mate or anything like that. So our model turned out more realistic. So I'm aware of wanting to leave some time for questions. So I probably don't want to spend too much time here. The main thing I just want to highlight is that effect where Wright-Fisher can actually strongly mislead you, and the maximum likelihood models, if you learn selection and dominance, can be way off the truth because they're sort of overconfident and they're overcorrecting for drift. And in terms of limitations, it's always going to be hard to tell apart selection and drift. We saw some examples of that, and people argued about it for, decades. There's some very recent methods that were discovered kind of writing up the paper that we haven't benchmarked against. So there's some possible more work to be done there. But we're probably just going to try and submit the methods as they are and see what reviewers say for that. And yeah, we can't infer the population size with our method, but there are other ways to estimate it independently. And I believe that's all I wanted to say. Thank you for listening. I went a bit longer there than I intended to, so sorry about that, but I'm happy to take questions with however long we got left. I think it's, yeah, thank you.